Hello students, you are welcome to the digital learning platform of Edusat Odisha. In the previous classes, we discussed about the genetic material, the structure of a gene, and very recently replication of a DNA, the mechanism by which DNA replicates. Towards 1958, Crick proposed central dogma of molecular genetics. Central dogma, what does it mean? It is a central phenomenon, it is a concept which explains the flow of genetic information from cell to cell by means of replication. Okay. Today, we will be discussing central dogma of molecular genetics, transcription and the role of RNAs in protein synthesis. So, let us come to the central dogma which was proposed by Crick in 1958. That concept explains how information flows from DNA to DNA by means of replication. Okay. So, that is generation to generation information flow by means of replication, but how information flows from DNA to protein, that is within a cell. DNA gives rise to RNA, RNA gives rise to protein. So, the process of information flow from DNA to RNA is your transcription, from mRNA to protein, that is your translation. The two processes, transcription and translation, explains how information flows from DNA to protein. So, this is the unidirectional flow of information from DNA to protein, and DNA to DNA by means of replication was explained by Francis Crick, but subsequently with advancement of a technology, these retroviruses were discovered. They have single stranded RNA as genetic material and a unique enzyme called reverse transcriptase, that is an RNA dependent DNA polymerase, which use RNA as template and synthesizes DNA. Okay. So, this was for the first time the enzyme was discovered which reverses the transcription, that is why the name was reverse transcriptase. So, with the discovery of this reverse transcriptase in retroviruses, the central dogma was reorganized, and you can see here the solid arrows shows the original central dogma proposed by Crick, but subsequently the dotted arrow explains that how RNA give rise to double stranded DNA. So, this is the present form of central dogma. So, this Howard Temin and David Baltimore, their discovery of reverse transcriptase gave a new form to the central dogma, but subsequently reverse transcriptase became a tool in genetic engineering for synthesis of complementary DNA. We will see later. So, now prior going to the process of transcription, let me tell you why it is so called. Some fundamentals must be understood before going to the process of transcription. So, transcription is a process from which DNA give rise to RNA, DNA of a gene give rise to RNA, but DNA and RNA both are written in the sequence of bases. They are a language, that is nucleic acid language, but two versions, two scripts. One is DNA script, that is written in ATCG, another is written in AUCG, but both are nucleic acids. So, only script is changed. So, the process by which DNA gives rise to RNA by means of transcription process. Okay. So, this is the first step of information flow within a cell. So, now let us understand what a gene is. Certain things I want to explain, that a gene is really transcribed. Gene is really transcribed. So, transcription, as we understood the two terms transcription and translation, let it be clear. The terminologies are important. So, transcription, DNA give rise to RNA, but DNA is written in ATCG sequence, but it is written in AUCG sequence. Only the script is changed, that is why it is called transcription. But in case of translation, what is in that the sequence of bases of mRNA, are converted to the sequence of amino acids, So, the language is entirely changed. So, that is why it is called translation. Okay. But the details of which will be explained later. Now, what is transcribed, whether it is DNA is transcribed or a portion of the gene is transcribed. As we know, genes constitute hardly 3% of the entire genome. The genes are transcribed. The DNA of gene, a gene is transcribed. Okay. So, here if it is at all a gene, it is a stretch of DNA in the chromosome which has a definite boundary, but in general DNA has two regions, especially a structural region, the other one is the promoter, that is the regulatory region. So, now here I can say you it is the structural region which is transcribed, this region is not transcribed, but this regulatory region helps the accurate transcription of this structural region. So, then let me explain the major difference between prokaryotic and eukaryotic genes, because prokaryotic genes are continuous genes. At least during our first year syllabus, we must have encountered the terms monocystronic and polycystronic genes, So, prokaryotic genes are polycystronic, but this is the case of eukaryotic gene. So, eukaryotic gene are monocystronic, but prokaryotic genes are polycystronic. What does it mean? A single gene here, in case of this, a promoter controls the transcription of a single cistron structural unit, but in case of prokaryotic system, many cistrons, many structural regions, if this is gene 1, gene 2, then this is gene 3, then this is the promoter. So, in case of a prokaryotic system, many genes are under the control of a single promoter and they are polycystronic and they are organized into operons. So, the prokaryotic genes, many structural units are under the control of a single promoter, that is why they are called as polycystronic. Further, you must be remembering from the first year that each cistron or the structural region in prokaryotes is continuous, whereas the cistron in eukaryotes is discontinuous. It is having exon and intron, likewise. So, in case of eukaryotes and prokaryotes, the major difference between the structures is that it is monocystronic and cistron is split. So, that is why it is split gene, but these are continuous genes. Now, question comes, a gene is transcribed, there are two strands of a gene, but what strand is transcribed? Now, I am describing that there are two strands of a gene, and if it at all, this is the promoter region and this is the structural region, there are two strands of a gene, but whether both the strands are transcribed or one is, exactly only one strand which is fixed for a particular gene, that is transcribed. If both the strands will be transcribed, different kinds of proteins may be produced, but initially if this strand and this strand, if they are transcribed, they will give rise to two different types of RNA which are complementary to each other, and they will pair up and will be degraded. It will be of no use. So, now the strand that is transcribed, let me tell this strand, because the template strand is runs from 3' to 5' direction, and this is the RNA chain. This is the RNA chain. RNA, like DNA, is synthesized from 5' to 3' direction, but template is transcribed from 3' to 5' direction. The strand which is transcribed is called template strand. Okay. So, this is called non-template strand, non-template strand. Here let me tell you, this non-template strand is your coding strand, coding strand. It seems very odd, because this is giving rise to RNA, but how can this called be coding strand? Because this RNA has the sense for protein, it codes for protein, as this strand is equal to this strand excepting thymine and uracil. That is why this strand is called coding strand, and this strand is also called sense strand. The motive behind drawing this to explain that this one is called sense or coding strand, the template strand from which transcription occurred, that is your antisense strand, antisense strand. So, antisense strand is really the strand which undergoes transcription, not both the strands, only one strand undergoes transcription. Now, look at the screen. Here I have given just an operon in which 3 genes, gene 1, 2, 3, they are contiguous, pakha-pakhi heiki achandi, and they are the under of control of a single promoter. This is the organization in case of prokaryotes. Now, because in our syllabus transcription in E. coli or prokaryotic system is mentioned, that should be explained well. So, now a gene, a regulatory region which is called promoter, and the structural region which undergoes transcription. So, the template strand of the structural region undergoes transcription. So, now the 3 regions of a gene, because a gene has a boundary whether it is in prokaryote or eukaryote, gene meant for function. A gene has a promoter, regulatory region, where from the transcription starts, or that directs from where the transcription of the gene will take place, and ultimately another sequence towards the end, that is downstream of the coding sequence, is your terminator. That sequence has the signal or has the sequence which directs termination of the process of transcription. Now, my question is, promoter is a regulatory region that is upstream of the coding sequence, that is towards the 5' end of the coding sequence, present towards the 5' upstream of the coding sequence. Now, another thing is how the enzyme which carries out transcription, that is RNA polymerase. In prokaryotes, there is a single type of RNA polymerase. It transcribes all kinds of, or it synthesizes all kinds of RNAs, tRNA, rRNA and mRNAs. It has only one type in its cytoplasm, in its cell, but prokaryote has, E. coli has about 4,500 genes, but how the enzyme being the same, how it identifies what gene at what time is to be transcribed. Here I want to mention 2 important things. the enzyme has a sigma factor, I will come to that. That sigma factor recognizes the promoter. So, promoter is important. So, promoter of all the genes in prokaryotes when sequenced compared, when they are compared, it was found that upstream, that is towards the 5' end towards the -10 base sequence, there is a consensus sequence. There is a consensus sequence which is hexanucleotide T A A T A T. So, towards -10 region, towards -10 region, this Pribno box is common to almost all the genes. That is why it is called consensus sequence. This sequence is identified by the sigma factor of RNA polymerase. Here let me explain what +1 and what -1, -2 and -10. +1 is the base, the first base which is copied, transcribed to mRNA, that becomes the first base of the mRNA, and minus is the base that is just upstream of that. So, between +1 and -1 there is no gap, no 0. So, +1 is the first base to be transcribed, -1 is the base upstream, first base upstream of the promoter. So, prokaryotes have the Pribno box. Similarly, eukaryotes towards -25 region they have a Tata box called Hogness box. So, promoters are essential for transcription process. Why I emphasized this? Because you must know eukaryotic genes, insulin gene is now placed in bacterial system. So, when this is placed, it is the insulin gene coding region by structural region, it must be tagged to the prokaryotic promoter, otherwise it will not work or operate, not be transcribed in the prokaryotic system. So, understanding the promoters, structural regions, property of the structural region is very important, while we will be going to genetic engineering in the subsequent sections. We will be discussing transcription in Escherichia coli, a typical prokaryotic cell. In prokaryotic cells, there is a single RNA polymerase, that is called DNA dependent RNA polymerase, because that uses DNA as template and synthesizes RNA. That is why it is called DNA dependent RNA polymerase, and it is only of one type in prokaryotes. So, let us have a look, because on this, on the structure of this E. coli RNA polymerase, because it is very important in the process of transcription. So, this consists of 5 different polypeptides of 4 kinds: alpha 2 molecules, beta, beta dash and sigma. The first 4 constitute the core enzyme and the association of the sigma factor, because sigma factor is dissociable from that. Okay. So, the core enzyme, when the sigma factor associate with that, that becomes the hollow enzyme. So, the function of the hollow enzyme is it recognizes the promoter. In experiment, it is seen that when only the core enzyme is given to the DNA template and RNA of different segments or different lengths are formed, but when the hollow enzyme, that means core plus the sigma factor in toto given to the template or to the gene, it yielded a different RNA, which is very accurate one or desired one. That means, core enzyme alone cannot initiate the RNA chain in a definite point. So, for the accuracy from where the transcription would begin, it is the function of the hollow enzyme. So, the hollow enzyme is responsible for recognition, binding to the promoter and unwinding of the 2 DNA strands, because the DNA in double helical form cannot be copied. When it is unwound, when the two strands are separated, the bases are exposed, one of the two strand, that is the template strand, which is antisense DNA strand, that acts as template. So, the template is read from 3 dash to 5 dash direction, transcribed from 3 dash to 5 dash direction. Another major difference of the RNA polymerase from that of DNA polymerase is that it initiates the chain de novo. It does not need any primer as the DNA polymerase 3 was doing. So, now the another important feature of prokaryotic transcription is that the building blocks are ribonucleoside triphosphates. They are ATP, UTP, CTP and GTP. So, these precursors, they come to the site of the enzyme as triphosphates, but what remains in the RNA chain, that is the monophosphate form. What is hydrolyzed, what is cleaved? Now, that is the pyrophosphate. That pyrophosphate, that means PPI, is the source of energy. The phosphate phosphate bond, when it is hydrolyzed, that provides the driving force for the polymerization process. So, the nucleoside triphosphates, they are working as the building blocks side by side, they are also the source of energy for the reaction process. Okay. DNA is transcribed in 3 dash to 5 dash direction, but RNA grows from 5 dash to 3 dash direction. Now, the process systematically can be studied like this. The process of transcription can be studied under the headings, that is the binding of RNA polymerase to the promoter of the template, then initiation of the chain, elongation and chain termination. So, initiation is brought about when the RNA polymerase binds to that. I have explained well how the hollow enzyme binds to the consensus sequence of a promoter in gene. So, now you look at this diagram, the sigma factor and the core enzyme, total the hollow enzyme that binds to the -10 base to -35 base sequence. So, that these are the consensus sequences among different genes. Okay. So, that helps in the binding of the core enzyme, hollow enzyme to the promoter. Once recognition, unwinding and initiation is over, then the sigma factor dissociates, because that sigma factor is loosely bound to the core enzyme. So, now in step A you see how that associates, then initiation takes place from the +1 site, then the sigma factor goes away and then elongation starts. So, what is seen here, I can say there is a temporary local unwinding of the base. What long of the DNA is unwound? It is exactly 17 nucleotides long DNA is unwound locally by a RNA polymerase for transcription to take place. It forms a transcription bubble. If you look at, it is a transcription bubble. There the unwound DNA, the growing RNA chain, they constitute the building blocks, the enzyme, everything constitute the transcription bubble. So, now in the elongation phase, RNA synthesis begins after the formation of the first phosphodiester bond, whether it is DNA or RNA, does not matter. Everywhere nucleotides are linked to each other by means of phosphodiester bond. But what would be the first base, what would be the second base, that what would be the third one? It is not arbitrary, it is directed by the complementarity of the template strand. So, template strand determines what would be the sequence of bases in RNA. So, an important change in the elongation phase is the dissociation of the sigma factor. Then the transcription bubble, as I have already discussed, it moves downward. Why downward? Because in the upstream there is promoter, in the downstream it is a coding sequence. It goes towards by way of a polymerization. The transcription bubble gradually moves towards the transcription termination sequence by addition of nucleotides. If you compare DNA replication with transcription, 800 nucleotides are added per second in DNA replication, whereas in transcription, the RNA chain elongation takes place about by addition of 50 nucleotides per second. So, as the DNA bubble moves, the DNA unwinds ahead, because it has to open up the strand, make available the template, but on the behind it must rewind. So, the basic reaction carried out by RNA polymerase is like this: a particular length of RNA having n number of nucleotides and one nucleoside triphosphate. In each time, in each reaction, there is addition of one nucleotide, and this addition is made possible, or bond formation is made possible by means of a phosphodiester bond. So, this is a sequential growth of the RNA chain. Hope you have understood this well up to this. In continuation to the transcription process, let us look at the elongation phase, and a special discussion must be made of the transcription bubble. As bubble, we know a locally unwound two strands of DNA, and where you will find the entire region, the transcription bubble comprises of the locally unwound DNA, the nascent RNA chain, and the nucleotides, all constitutes the transcription bubble. It is almost 17 nucleotides long, 17 nucleotides long. So, when this elongation process goes on, it continues, but where it is to end? The gene has the signal for that, that is the termination sequence. As promoter gives the signal where to start, termination is the sequence, it gives the signal where to end transcription. So, it is there in the gene. So, therefore, I told you gene has a boundary, beginning and end, and different regions, two regulatory regions, promoter and terminator, and the coding sequence. So, now when the termination reaches, how termination is brought about? In the gene itself, the transcription termination sequence consists of a GC rich region, GC, GC, GC. Likewise, similarly, in RNA it would be CG, 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 followed by U, U, U in RNA. So, what would be there? Now, this RNA, because of a CG, CG, CG basis, it forms complementary structure, complementary exist. So, it goes for a secondary structure, which is a hairpin like structure. If you look at this structure, this looks like a hairpin with a head, and that is one unpaired region, and the stem having CG, GC, GC region like this. So, when in the RNA itself a stem hairpin like structure is formed, followed by U, 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 that gives a signal to the RNA polymerase, and the RNA polymerase halts, pauses therein, and after some time the U, U, U region also signals, and it there the RNA forms weak bonds with the DNA, and there the RNA terminates its synthesis. So, RNA releases, and the enzyme also leaves the termination sequence, as a result of which the hairpin like structure, because its conformation, three dimensional conformation, results in termination of the process of transcription. But in other cases, another factor called rho dependent termination, another protein is also involved in certain other cases, that I am not going to discuss, but rho protein is also involved in transcription termination. But when we are going to write these things in examination as a long question, a diagram, a running diagram must be there. So, you look at the three different steps: initiation, elongation, and termination. Sequentially they are given. So, I think what I taught you earlier in different phases this can be summarized here. RNA polymerase binds to the holoenzyme binds to the promoter, then once the transcription begun, the sigma factor dissociates, core enzyme catalyzes the chain, and when termination sequence is reached, then termination occurs. So, by this transcription in in prokaryotes came to an end. Hope by this process the RNA is released. Now, let us look at this diagram. So, it is transcription and translation in prokaryotes and eukaryotes. What I want to impress you? Transcription prokaryotes is a single compartment and eukaryotes are a multi compartment cell. Compartment of compartments is your eukaryotic cell. So, transcription and translation in prokaryotes takes place in the same compartment. As you can observe, while transcription is going on in prokaryotes and the 5' end of mRNA is available, there the ribosomes attach. Ribosomes begin translation. So, what I mean to say, in the same compartment simultaneously while synchronously while transcription is going on is still not finished, at the 5' end the ribosomes attached and bring about translation. So, this is called simultaneous or coupled transcription and translation in prokaryotes, but in eukaryotes it is quite different. All the transcription processes, all types of RNA are synthesized in the nucleus, because the genes, the DNA is housed in a compartment that is nucleus, but we know ribosomes, they are available in the cytoplasm for protein synthesis. So, the protein synthesis compartment is cytoplasm, but RNA synthesis compartment is your nucleus. In terms of space, in terms of time, transcription and translation are separated in eukaryotes. That is why it is called spatially in terms of space, temporally in terms of time. Processes like transcription and translation are taking place in eukaryotes. So, as it takes long time between transcription and translation, there are many regulatory steps in eukaryotes as compared to prokaryotes. Now, we learned a lot about, or we discussed a lot about transcription in prokaryotes, but what about eukaryotic transcription? In eukaryotes, transcription and translation are separated in terms of time, that is temporal separation, in terms of space. So, that is called spatial separation. But we discussed a lot about prokaryotic ones, but eukaryotes have a lot of differences between in their transcription process from that of prokaryotes. Here I will explain first the structure of eukaryotic gene. We know eukaryotic genes are split gene. Here simply I want to write, this is the promoter and this is the coding sequence. This is the coding sequence CS. The coding sequence has exons and introns. Here I will write exon 1, this is intron 1, this is exon 2, this is intron 2 and this is exon 3. So, these exons and introns, what does it mean? Exon means, it must be well understood, exon expressing region. So, these exons are the regions or the sequences exon. These are the regions which must be finally reflected, these must be finally spliced back to form the functional mRNA. So, these exons have the information for messenger RNA and subsequently proteins, but what the introns are? Introns, where the term comes, introns. So, these are intervening regions, intervening regions. So, from these two terms, these int are introns. So, introns intervene the continuity of the coding sequence. So, that is why they are called as introns. I may tell you this is the nature of the split gene. Split gene was discovered in 1977 by Philip Sharp, okay, Roberts and Philip Sharp in 1977 in adenovirus, which is a eukaryotic virus. So, that led to the discovery of split gene of eukaryotes. So, now question arises, when this RNA is transcribed, DNA is transcribed to form RNA. Okay. When this is transcribed, because this much is not going to be transcribed, this is to be transcribed. So, this is your primary RNA transcript, primary RNA transcript. The product of transcription is called transcript. So, here like DNA, it has also introns, it has also introns. So, these introns must be removed, must be removed. Okay. I will show how these introns removed and this transcript is processed. I will show you this in the slide, but here let me emphasize that in contrast to prokaryotes, eukaryotes have 3 different types of RNA polymerases. They are RNA polymerase 1, RNA pol 1, in short polymerase 2, polymerase 3, but prokaryotes have only one type. Prokaryotes have only one type. So, RNA polymerase 1 give rise to rRNAs, rRNAs. This give rise to mRNAs, which give rise to proteins, and this give rise to tRNAs. So, I have remembered in my life like this 1, 2, 3 RMT, RMT. So, ribosomal RNAs, this is 28S rRNA, okay, 18S rRNA and 5.8S rRNA. All RNAs, mRNAs are synthesized by this, and tRNAs and 5S RNA by this. So, there are 3 different types of RNA polymerases to synthesize different types of RNAs in eukaryotic cell. So, now these can be distinguished or this could be discovered because of one poisonous mushroom, one toxin, Amanita phalloides, Amanita phalloides, which is generally called death cap mushroom. It has one active component called alpha amanitin, alpha amanitin. Its application led to the differentiation or distinguish the 3 different kinds of RNA polymerases. This is insensitive to the alpha amanitin. Polymerase 2 is fully sensitive and polymerase 3 is partially sensitive. So, this differential sensitivity of RNA polymerases to alpha amanitin led to the discovery of 3 different kinds of RNA polymerases. So, now I can tell you these 2 are present in the nucleoplasm. Where they are located? Nucleoplasm. But can you guess where this is? Because the little nucleus, that is nucleolus, is the site of ribosome assembly that carries the nucleolus organizing region. Therefore, this RNA polymerase is localized in the nucleolus. So, this is a nucleolus localized polymerase, but these 2 are present in the general nucleoplasm. Now, let me explain how this RNA processing occurs. So, the immediate product of transcription is transcript. It contains both exons and introns. The processing of primary RNA transcript is very complex in eukaryotic system. As part of the eukaryotic processing, there are 3 events. one is called capping. You will see, as you see, the first end or first terminus to emerge of the RNA is 5' end. This 5' end is capped by addition of a 5-methyl guanosine group. So, capping is addition of 5-methyl guanosine, a chemical group, to the 5' end, and once transcription is over, then template independent polyadenylation takes place towards the 3' end. You see, once transcription is over, in the green color a polyatel, that means many adenosine residues, adenylic acid residues are added, mostly 200 to 300 residues are added to the 3' terminus, but that is not template dependent, that is template independent. So, capping and tailing. Side by side, removal of introns takes place by looping mechanism, they are looped out and chopped, and the exon 1, exon 2, exon 2, exon 3, they are joined, spliced back. So, as a result of which removal of introns and joining back of exons makes the exons continuous, as a result of which the functional mRNA, the matured mRNA is too small as compared to the primary RNA transcript. So, this difference in the length of or the size of the primary RNA transcript and the matured mRNA led to the discovery of this mechanism. Okay. So, the messenger RNA is too short in comparison to the primary RNA transcript. Here I want to mention one term called heterogeneous nuclear RNA, heterogeneous nuclear RNA. Here we see there are 4 different RNAs of 4 different lengths. So, this population of the RNA transcripts at different stages of development constitute a population, and that population is called heterogeneous nuclear RNA. So, this is the specificity of eukaryotic transcription. Now, let us go for the details of this process. The splicing process must be very accurate. Splicing involves removal of introns and joining back of exons. If a single nucleotide of exon is retrieved, then the mRNA would be harmful. It will not give rise to a right kind of a protein. So, it must be a very accurate process. So, the machinery which brings about this splicing mechanism, it is called spliceosome, and here by experiment it is seen that the RNA, primary RNA transcript is self splicing, it is catalytic in function. The RNA along with other proteins is responsible for this excision and joining. So, therefore, the spliceosome is a ribozyme. Here I can say here the RNA itself acting as an enzyme for its splicing mechanism. So, up to this eukaryotic transcription and its specificity, for example, uniqueness, I think you people have understood well. By transcription, messenger RNA is formed. Okay. So, a protein encoding gene on transcription give rise to mRNA. Similarly, the genes for rRNA, genes for tRNA are there. So, gene can give rise to tRNA, protein encoding gene can give rise to mRNA, rRNA encoding genes can be or they can give rise to, by way of transcription they give rise to rRNA. So, here once this mRNA is ready, it is a genetic RNA. What the cell be doing with this? The mRNA must be translated. Translation, the second step of unidirectional flow of genetic information from DNA to protein, the second step, that is translation. Before the understanding the process of translation, we must understand the 3 different kinds of RNA. They are the intermediary molecules, they are the workers of the cell, they help, they realize protein synthesis. So, here in the context of protein synthesis, I am describing what the different kinds of RNA do. We know the three classical RNAs, one is ribosomal RNA, transfer RNA and messenger RNA. Ribosomal RNA, we see, look at this table, they by weight they constitute 80%, transfer RNA they constitute 15%, and the meager amount is your messenger RNA. And the size you see, their sedimentation coefficient 23, 16, 5 and 4. Transfer RNA they have a sedimentation value of 4s, 4s, they are very small. Because of their small size, they are soluble in the cytoplasm, that is why they are called as soluble RNA. Okay. So, now look at the number of nucleotides in them. Ribosomal RNAs 23s has 3700 nucleotides, 16s RNA has 1700, likewise it diminishes. And most important, we know the transfer RNAs they have in average 75 nucleotides in them, okay. And all the RNAs are single stranded, DNA is double stranded, but being single stranded, do these RNAs work? No. So, here only we will discuss how they are organized in 2D or 3D, what is their native structure in the cell and what they do. Let us look at this. Let us look at this bacterial messenger RNA. This is divisible into three parts. In the center, there is the coding sequence which codes for the protein to be synthesized, and in the beginning of the coding sequence, there is a start codon that signals protein synthesis to be start with, and at the end of the coding sequence, there is a stop codon and that signals that protein synthesis would be stopped here. And in the upstream of the coding sequence, there is a 5' untranslated region. As the name suggests, it is not translated, rather it has a ribosome binding site. So, during protein synthesis, a ribosome sits in this ribosome binding site and then it starts protein synthesis. And at the 3' end of this coding sequence, there is a 3' untranslated region. So, both 5' untranslated region and 3' untranslated region has regulatory roles. This poly-A tail is variable. Now, messenger RNA has a very short lifespan, and in the messenger RNA, message is written in the sequence of bases. Very shortly, or in the coming class, we will be coming how the bases are read. The bases of this messenger RNA, which is a sequence of continuous bases without any gap, gap or a punctuation, they are read continuously 3 at a time, that is 3 consecutive bases makes a codon, and that is read by your transfer RNA. So, next is your here the messenger RNA has certain features, they contain a continuous sequence of nucleotides encoding specific polypeptide. One mRNA give rise to a specific polypeptide, they are found in the cytoplasm, because the site of protein synthesis is your cytoplasm. They are attached to the ribosomes when translated, that means ribosomes are the macromolecular complexes, they attach to the messenger RNA, then upon which the transfer RNA binds to bring about a translation. So, this is a complex interaction of 3 different kind of RNAs to translate or to bring about protein synthesis. So, besides this coding sequence, the untranslated region at the 5' at the 3' end, they have regulatory role and they also contribute to the stability of messenger RNA. Here, besides the messenger RNA, which is genetic in nature, that carries the genetic information, the transfer RNA, which have a very small value, 4s value, its nucleotide length varies from 75 to 95 nucleotides. It has a linear structure, it can give rise to a secondary and tertiary structure. in tertiary structure you can see it has two ends. Its name is transfer RNA. What it transfers? If I make this as the ribosome to which the messenger RNA binds, and this is the cytoplasmic area. So, transfer RNA binds the ribosome amino acid and brings it to the site of protein synthesis, that is the ribosome. So, that is why its name is transfer RNA. It transfers amino acids from different corners of the cytoplasm to the site of protein synthesis, that is ribosome. So, it has two different important points. One is anticodon site, the other one is amino acid binding site. In the two arms, opposite arms, in one end it reads the codon through its anticodon, and in the other region it binds the amino acid. Okay. So, transfer RNA are very important in the translation process. And the third one is your ribosomal RNA. They constitute the major component of the cellular RNA, but ribosomal RNA they do not work singly. In association with many number of polypeptides, they give rise to ribosomes. So, ribosomes are macromolecular complexes. They are molecular machines, I will explain in the coming session. So, here any ribosome comprises of two units, one is the small subunit, the other one is a large subunit. In case of bacteria, this is small subunit is 30S and large subunit is 50S, and in this subunit form they are remaining in the cytoplasm, but when they come to protein synthesis they assemble, that give rise to the 70S ribosome. So, ribosome helps in translation, it reads the mRNA, and one thing you remember, the rRNA 23S rRNA or 28S rRNA present in the large subunit is the peptidyl transferase. It brings about peptide bond synthesis during protein synthesis. To sum up this process, I may say you the genetic information flows from DNA to messenger RNA, from messenger RNA to polypeptide, and the first step is transcription and the second step is translation. So, this is the unidirectional flow of genetic information to realize protein synthesis. Why these things must be explained well? Because the ribosomes, unless we know the different sites of this ribosome, we cannot target this. So, ribosomes are targeted by biochemists for development drugs. Okay. So, now I hope you people might have understood well the process of transcription, prokaryotic, eukaryotic, their difference, uniqueness, and this will help us in understanding the second process of unidirectional flow of information from DNA to protein. The second step is translation. Hope we are in a position to go to translation in the coming class. Thank you.